Looks like it's time to begin. So the good news is this will be our last lesson that is primarily focused on scientific data. So for those of you that are kind of overwhelmed by all this sciencey stuff, this is the last one that's going to be primarily focused on that. The rest would be archaeology. That being said, this is probably one of our more in-depth ones, so I'm sorry about that. We're going to be talking about the animals today that were living in this post-flood ice age time period. As far as men, we'll be focusing on them next time, but for animals, we'll be focusing on them today. So the question is how after the flood did animals get where they are, and how do we get the today's animals, since we know that the animals on the ark didn't look like today's animals, since today's animals we have so much variety. So, a number of ways they could have done that. Flying, walking, swimming, or floating along mats. Flying and swimming are probably the easiest because they could still get to where they need to go. But walking, as we talked about last time, at the peak of the ice age, a lot of water was on top of the continents. We had all those lakes, those humongous lakes, as well as ice sheets that were thousands of feet thick. This allowed for a number of land bridges to be formed to America, Japan, England, even Australia, as you can see in that picture there. And a number of animals likely were spread by humans. We see that even up to near modern times, humans taking animals wherever they go. Deer, boar, pheasants, rats, etc. Sometimes intentionally, sometimes unintentionally. And it's even been suggested that all the Australian dingoes, the wild dogs in Australia, have been descended from a single female domesticated dog from Southeast Asia. Well, another interesting fact is about Australia is that plus America are the only place that are inhabited by marsupials, which is interesting since those are the farthest places from the Ark, where the Ark landed in Mount Ararat. But it makes sense since marsupials, since they carry their young in their pouch, their pregnancy, their gestation usually is easier as far as ending it, since their young are very, very small and finish developing inside the pouch. They don't have to slow down to produce offspring. They could travel much, much faster. And this environment would have been a very dense environment, as we discussed last time. With all this moisture, with the sediments, all that earth having a lot of volcanic ash, as well as a lot of organic material from all the dead things in it, would have been very lush for plant growth. We've had a lot of forests at this time, meaning that the animals then could thrive. When you have a small amount of competition, they would have just went to an area with no competition. Thus, they would have continually spread out, going from little to no competition areas. Eventually, things would have filled in closer to today. But for a few hundred years, they would have just been spreading out pretty quickly. But let's talk about those floating log mats. Rafting is a common way of which things can go over water. We do it today as humans anyway. But the question is, would animals have done this? And would there have been these mats existing post-flood? Well, let's look at tortoises. Tortoises are found all over the world, on all continents and many, many islands, yet they cannot swim. They're different from turtles that can swim. Tortoises cannot. So how did they get everywhere, including these islands? Because even though there would have been a lot of land bridges, not all islands would have been connected by land bridges. Well, we see animals doing this today. We see green iguanas rafting to certain islands today. And even the evolutionary model requires this method of transportation. They use that to get lemurs where they are. So at the end of the flood, there would have been a lot of logs from all that destruction. We've seen today with Mount St. Helens, which is a very minor catastrophe, it blew a number of logs down into Spirit Lake. And these logs stayed there for decades. In fact, they're still there. Based upon the measurements they have taken, the log mat in Spirit Lake has a half-life of 75 years, meaning that it takes 75 years for half of those logs to sink out. So that means this log mat in Spirit Lake likely exists for over 100 years, 100 plus years. And this is a very small catastrophe. While a good portion of all that vegetation pre-flood would have been buried in the sediments producing coal, natural gases, and stuff like that, it's reasonable to believe that a good portion of them would have survived in these turbulent oceans, collecting and basically making these log mats. And this would explain why we have something called an island effect. Today we see creatures on islands that are either giants or dwarfs of their kind. such as the giant tortoise, only found on islands, and the dwarf elephant, found on islands. This is very peculiar, but it makes sense with the floating log model. Basically, these smaller ones, like the smaller tortoises, cannot as easily grab upon a log because they can only fit on one log. If you're just riding on one log, it's very easy to roll off. Same way for elephants. Modern elephants are pretty big. They've been even bigger in the fossil record. It's very hard to have something that heavy, that big, float on just a pile of logs. It's easy for them to fall off. But a certain size, about the size of the giant tortoise or a dwarf elephant, they can grasp and latch on to a number of logs, 2 logs at the same time, which is very difficult for it to fall off, or less easy for it to fall off, I should say. And these log mats would have floated on these currents you can see on the picture there. And it makes sense that we have giant tortoises in the Indian Ocean on an island there. You follow the currents and it goes right by Galapagos through the Pacific. And so those are the 2 places that we have giant tortoises. And it seems that a giant tortoise got on and rode around. Now, it's hard to prove this, but it does make sense. Another problem that people have with the biblical model is how do we get all these animals post-flood? In the peak of the Ice Age, we have a lot of dead animals. We talked about all those extinctions. We have millions and millions of animals, all dead in the permafrost. So we know that these all were existing about the same time. So how do you get millions and millions of animals in a few centuries? Especially since elephants are one of the slowest breeders, and we have about 50 million mammoths frozen in the permafrost. Now, elephants take about 10 years to mature, and they produce 1 offspring at most every 2 years. In Africa today, in a preserve, in some preserves that have good management, elephant populations can double every 10 years. And post-flood, we're expecting a very lush, very fertile environment, environment very low in competition, meaning that the growth rate of these species would have been very, very high. And if you do all that math, it accounts for billions of elephants in a few hundred years. So it makes perfect sense. So there's no real objection scientifically to what we're seeing in this post-flood world. But the question is how do we get from this post-flood world to animals today? How do we get all this speciation? Speciation is the development of new species. Basically, not evolution as you think of it, but simply the biblical kind separating out into different species, such as breeding of dogs. So many of us are aware that in the not too distant past, we have taken some sort of canine and produced all of the current breeds from that small section of canines by selective breeding, by removal of certain genes, and selecting for what we want in a dog. Now, something very similar would have happened in the post-flood world. And we'll get into how and why we think this is the case, based upon genetics and some historical facts. But first, we need to compare a species versus a biblical kind. The Bible gives us one real definition for a biblical kind, that they can produce after their own kind, so that they can breed. So we know that if animals can breed, they're of the same biblical kind. So if you look up at the top right, you can see a giant tortoise, and then some other tortoises, and then a turtle, and then a snake. Based upon this simple breeding characteristic, it seems that most of today's modern taxonomy, at the family level, that would equal the biblical kind. Not always, though. Some would be at the genus level, but a good portion, majority, would be at the family level. Meaning that the tortoises can breed, but they cannot breed with the sea turtle or the snake. So breeding is the primary way we differentiate the biblical kind today. But there are other ways. Because some animals can't breed. Take cats, for example. You cannot have a house cat breed with a tiger. But lions and tigers can breed. Cats, bobcats can breed with lynxes. And you basically can connect all of the modern cat species together by breeding from the smallest to the second smallest to the third smallest and so on. To the point where you can have all of them interbreed. So this shows that even if they cannot interbreed today, they can still be the same biblical kind. And some interesting facts about the cats is that the cheetahs, they are most closely related, meaning they can interbreed with pumas. Cheetahs are found in Africa. Pumas are in the Americas. And that's interesting because that means that cheetahs, whatever kind of cat you want to call it that was on the ark, migrated over to the Americas, produced the puma population, migrated back, and then sometime on their migration back, they got into some sort of catastrophe. They lost a lot of their population. Most likely, flood, volcanic eruption, all those horrible things that are occurring post-flood world that we talked about last time, probably killed a lot of them, meaning that there was a small population of cheetahs. And we know that because today, cheetahs are all basically equal to cousins. They're not great genetically. They have a lot of problems, even though, arguably, I think at least, they're probably the best big cat you could have. But that's my opinion. And so, if we have all these cats that can interbreed, but some that can't interbreed amongst themselves, though you can get a connection, we can get more information as far as what is a biblical kind. We can also use something called cognitum. I can't say that very well. But this is using morphology. So since we know that a small cat and a large cat look similar, very, very similar in fact, there's a good chance that they are the same biblical kind. And we know that they are based upon those breeding patterns we talked about. We can then use that comparison to extinct animals that we only find in the fossil record, and then use that to loop them in. Now this is by all means a less accurate method of determining biblical kind, so we don't like to rely on this. But we're showing that there are other ways besides making sure that two animals can breed today, since there have been some changes. The question also comes, what if they can interbreed, but their offspring are not fertile, such as the horses and the donkeys? Because mules are infertile, right? Well, yes and no. We have actually seen mules produce offspring, not frequently, only about 60 times in recorded history. And this is because horses and donkeys have different number of chromosomes. A horse has 64 total, a donkey has 62, which means a mule between a horse and a donkey is going to have 63 chromosomes total. Having an odd number of chromosomes is not good, but they can function, they can do everything they need to do. But when they try to breed, you're not going to get a good one-to-one matchup on the genetic material of the father versus the mother, whether it's two mules or a mule and a donkey or a mule and a horse. But it can form that. And so we know from this that the number of chromosomes doesn't equal biblical kind either. And so we're getting a lot more information of what defines a biblical kind by going from what the Bible says and extrapolating that based upon what we're seeing today. So this means that if animals have different number of chromosomes, we cannot assume that they are not the same biblical kind. And this is something that is likely produced by a thing called a Robertsonian translocation. This was found out in about the 1916s and occurs about 1 in every 1,000 live births. And this is humans, not animals. It occurs when you have two chromosomes. And as you look in the bottom right, the two different colored chromosomes there. There's a large portion of DNA on one side of the centromere, and this is where they attach to and separate it out by the cell. And then on top of that, you have a very small amount of DNA. Well, something happens, and that small amount of DNA is removed, and a small amount of DNA on another one is removed. And then those two chromosomes attach and bind together, creating a new chromosome. So going from 2 to 1. And this likely occurred sometime in the donkey's lineage, since donkeys have fewer chromosomes than horses. Now they are going to be losing some DNA. And if this occurs and too much DNA is lost, then it is lethal, or it can produce a number of defects. But if it occurs in a very small amount of DNA that isn't necessary, remember, God put a lot of redundancy in our systems, then they can function and continue to produce. And based upon this, we also know that the same thing occurred in sheep and goats. Because if you have a goat and a sheep interbreed, which again is very, very rare, but possible, you're going to get a geep, or a jeep, depending on gif or gif. It's a geep, sorry, I just had to put that in there. But goats have 60 chromosomes. Sheep have 54. We know they can interbreed, though rarely. So that means they are the same biblical kind. And this means that a Robertsonian translocation must have occurred 3 times in its lineage to produce 60 to 54. So we're trying to get an understanding of what is in the biblical kind. So we know that there are a number of exceptions now. But let's dig a little bit deeper into the DNA and see what we can find that would help classify or clarify this. So we're going to get into the animal genetics. So a little bit of review. We already talked about how there's two types of DNA. First, the DNA inside the nucleus, which has most of our genes, which has the chromosomes in it. Now, based upon the large amount of DNA we have, it's very difficult to study. It's very large. There's a lot of differences in there. A lot of assumptions have to go into any study that focuses on this. So it's not great. But if we take a subsection of that and look at a single gene, such as the cytochrome C gene, we can see that animals fall into what are called nested hierarchical patterns, meaning you have one group here that's very similar, another group here very similar, another group here very similar, and another group here. Now the two extremes are the least likely, but all these groups do not overlap. They may be close, but they don't overlap. So that is why it's called nested. And while there are some sort of lineage, it's not an actual complete lineage, meaning that these groups are separated based upon their genes. Now, the evolutionary model would say that these separations based upon the DNA don't mean anything. They're going to be functionally neutral. They would not expect to have a different function in this gene here versus this gene here just because of a few minor differences. The biblical model would expect that in these nested hierarchies, the genes would have virtually the same function, because these nested hierarchies tend to align with the biblical kind. Now, outside those nested hierarchies, so we're comparing two separate nested hierarchies, then we expect that those genes would have different functions, because we are looking at a design feature. We're looking for evidence of design in there, and that's what we expect to see. So we're going to get back to that in a moment, but first let's rewind a little bit. So we cannot look at the whole amount of DNA to have any good evidence supporting one model versus the other, because if you look basically at mutation rates alone, you cannot get all the differences in DNA from that in 4,500 years. So that is why we, as the biblical model suggests, have heterozygosity not only in humans, but also in animals. So I just want to reiterate that. But the same problem is in the evolutionary model. They predict too many differences. You can see there, while we're dealing with big numbers, the low end of the standard deviation, that bar inside the bar, thin bar inside the thick bar, that is the standard deviation, meaning that is the potential numbers that could be accurate. It is still an order of magnitude, a number of orders of magnitude higher than what is actually seen today in yeast. Now, the explanation for both are a little different. The biblical model suggested that we have heterozygosity, meaning different genes, meaning God put two copies of genes inside the original creation. And so they had a lot of genetic diversity. Now there have been some changes from mutation, but very, very little over the last few hundred years. The evolutionary solution is that natural selection somehow reduced all those potential mutations into the mutations we see today, into the differences we see today. The biblical model has models that you can build upon that. The evolutionary model has no real mechanism for natural selection. We see a lot of mutations today that natural selection can't touch. And not just the neutral ones, but the ones that are actually bad. There's many genetic diseases today that natural selection isn't touching. It may be reducing those numbers, but it's still producing in that population, both animals and man. And so their model doesn't fit as well with what we see here, though the information doesn't match either model perfectly yet. And this is mainly because we can't get that information. There's too much of it to get to the fine-tuning understanding that we want. So we can't look at the DNA as a whole. That's the main reason why I want to mention this here. We have to look at subsections. And the best subsection is the mitochondrial DNA. Because, one, we know that there is going to be no heterozygosity in this, since we all just have one copy in our cells. The mitochondrial DNA is DNA inside the mitochondria of the cell, the powerhouse of the cell. And so we can factor out the heterozygosity and compare apples to apples with the evolutionary and biblical model with this. And again, the same expectations, biblical model, we expect some differences between biblical kinds, but no real difference among a biblical kind itself. And we still see a nested hierarchical pattern here. I cropped out the names on the left-hand side, but it's going to be the same as the top. The horses are going to be in the top left. Next, you have rhinos and tapirs, and then last, bighorn sheep. And so you can see the numbers there when you're comparing horses to horses. you're going to get a difference of about 1,100 base pairs or less. The less difference, the closer they are related. We're going to assume that. Now, you're going to get a big jump when you're comparing rhinos to horses with the next few columns over there. Over 1,900. Well over that in many cases. But when you compare rhinos to rhinos, again, you get a small subsection, another nested hierarchy. And the same thing when you compare tapirs and sheep to horses and rhinos, major differences. And so we're seeing this nested hierarchical pattern in both the nuclear DNA as well as the mitochondrial DNA. Again, this is not something that you would expect necessarily with the evolutionary model, but it is something you'd expect with the biblical model. That being said, it doesn't disprove the evolutionary model, it just doesn't support it. And when we look at the mitochondrial DNA based upon differences, based upon mutation rates, remember we're factoring out the heterozygosity. If we look at the evolutionary model, again, it predicts way too many mutations. Just doesn't fit with any of these. Now, we don't have a lot of mutation rates for mitochondrial DNA. It's really easy to get the actual DNA information, but they haven't done the studies yet to see the mutation rate. These are just some species that we have good evidence for that, good studies to show us the mutation rate, so we can get this study going. But when we compare the mutation rate to what we see today in the mitochondrial DNA, we can see that in all these species, it actually fits. It fits what we expect if these creatures were created 6,000 years ago on all of them. It's not a perfect fit for all of them. You can see there the fruit fly, the standard deviations overlap, but the averages don't. That doesn't matter. As long as the standard deviations overlap, then it's a good fit scientifically. I just want to point that out. We do have a number of other creatures' mutation rates. We have mice, some chickens, and a certain penguin. The problem is there's only one study for each of these. As far as science goes, one study does not a proof make. It's good evidence, but it doesn't prove it. You need multiple studies. You need to repeat it. But if we take these at face value, now the biblical model would predict 33, 6 to 158, and 55 to 168 for these species. The evolutionary model is next in that column, producing millions potentially, tens of thousands to millions of differences. What we see from these studies based upon the mutation rate, what we see in these actual mitochondrial DNA studies is that the difference is about 2 to 250, 2,500 basically, or less. So it doesn't match either model. But it's far, far closer to the biblical model. And if we do more studies, we'll probably get that number down. That's what we expect. But to my knowledge, we haven't had any new studies come out with this in the last few years. So we don't have all the information with this, but all the information we're getting is pointing towards the biblical model. So we went through how we can use things to differentiate biblical kinds. The nested hierarchies, the breeding patterns, certain morphology for the extinct creatures and stuff like that. And how the evidence, while not lining up perfectly because we don't have all the data in, but it is pointing towards the biblical model versus the evolutionary timescale. But can we go farther than these studies? Well, let's see. So let's look at how the mitochondrial DNA is going to affect speciation and see if we can get a clock from this. So today we have 70,000 vertebrate species, and those are creatures with backbones. They came from about 1,100 families. Now there would have been more families that were on the ark, more families of creatures, more biblical kinds. But we have a number of them that have died, and we found their evidence in the fossil record. But these are just the ones today. So how did 1,100 biblical kinds, 1,100 pairs of creatures on the ark, produce 70,000 species today? Especially since a lot of these were probably produced in a short period of time. Well, we know that this is possible since we produced a large number of breeds ourselves, both in dogs and horses and a number of other animals that we use today, cows and stuff like that. Horse breeders manipulate 10 to 28 million DNA differences when producing the breeds. This is a very small fraction of the DNA. And so a very small fraction produces all these differences, which is interesting. But we also have this in the wild population. Mice, for example. It is one of the most species-rich families, one of the most species-rich biblical kinds. There are over 700 documented species, which means that if we assume all these came from 4,500 years ago on the ark, That would mean one new species every 6 years. So let's see if we can compare this to any known historical sightings. And there is a verse in the Bible that many of you probably have just skipped over, Genesis 47:17. Many of you have probably just read through this and thought, okay, Joseph is getting rich here, good for him. But we see that horses and donkeys have already been separated at this time. This is 700 years, give or take, after the flood. Horses and donkeys are already separated. What's also interesting is that the flocks and herds are already separated. The word for flocks there is used for small cattle, sheep, and goats, which is very interesting since the genetic studies that we have today promote that not only donkeys and horses being the same biblical kind, but that sheep, goats, and cattle were once all part of the original biblical kind. They have separated out, producing a number of species, some of which can no longer breed together. But it is very strong evidence to say that this is the biblical kind, which it makes sense since this animal, all these are clean, all these have very, very similar exterior, interior, and these are basically one of the longest domesticated animals. And we have seen what we can do in the last few centuries. Imagine if we had millennia as a human race breeding these animals. So let's see if the mentioning here in Genesis 700 years ago, 700 years after the flood, can get a speciation event to produce these differences in that short period of time. So the stopping point we like to have is the present. We have to make some assumptions, so we're just going to assume that. We're going to assume that the starting point is based upon rooting at the midpoint there. And we're also going to assume that basically the mutation rate is going to be constant. We don't know that, but we're going to be assuming that it's a constant among all species in a biblical kind. Now we know there is going to be some variation, but we're going to assume that they're the same, just to make the math easy. Rooting on the midpoint is taking the two most different branches of DNA, two most different species, and then having the midpoint between them be the original section of DNA. Again, some assumptions in that, but you're going to get close with that model, that method. Ideally, we would have the mitochondrial DNA mutation rate, but we just don't have that yet. Hopefully in a few more years, maybe decades, we'll get that. So let's look at horses. We know that between horses and then donkeys and zebras, you're going to look at 24 to 27 million base pair differences. Donkeys and zebras are that much closer, and then the different zebra species being the closest of all. That's with nuclear DNA. So we need to figure out if the speciation events, the separation of these into different lineages, is going to match both the nuclear DNA, as we see here, with the mitochondrial DNA. And by doing that, we can see the same pattern in the mitochondrial DNA. So that means we can go from nuclear DNA and use the mitochondrial DNA for this study, since it's smaller and basically more able to get the information out of that. The problem with this, though, is that we only have 7 species of equines today. That's not great for statistics, but it is good for just a proof of thought, a proof of concept. So you can see here a mapping out of the equine speciation. You have horses breaking off first, then donkeys, and then finally zebras. Let's see if the same pattern holds with bovines. Now, in bovines, that's a family name. That is the biblical kind. There are over 100 species. With 75% of these species, the mitochondrial DNA has been sequenced. We know that because it's easy to get. And there are only a few thousand mitochondrial DNA differences between these different species. And so we're going to use the same rooting on the midpoint, all of those assumptions that we talked about. And we get something called a correlation coefficient that's very, very high. 0.9965. That's very high, meaning that there's a pattern here. And we'll get to that in just one second. But first, I want to show you this map.
Meaning what, John?
Meaning that there's a pattern here.
Okay.
Yeah, and we'll get to the pattern here in a second. But I want to show you all the different species that we get in this biblical kind. We have goats there at the top, followed by sheep, and then a number of species you probably don't know. The second half here, we have, again, a number of species we don't know. Gazelles, and then cows, bisons, and stuff like that. So basically, I went through and got some pictures. This means that all these animals here are the same biblical kind. And all these animals here are part of that same biblical kind. You can see that there are some differences, but they all have some similar features. 2 horns, unless we remove them with the cow there. Ears, nose, eyes. Internally, their digestive system. But there are a lot of differences. It's just shocking how much variety, how much information God put inside these biblical kinds, inside us as well. It just shows the grandeur, the wisdom, the intellect of God and his power. So I just wanted to take a moment to look at this and to just appreciate what our God made. Now, if we map out all those on a line here, you have mitochondrial differences, so basically a time elapsed in differences of mutations building up. Compared to the number of species on the y-axis, you get this nice straight line. And that's the pattern I was talking about, meaning that we have something that we can use to predict what's going to happen next. And if we match this to what we see in Genesis there, Genesis 47:17, we can get some evidence of a timeline and see if what we're seeing today matches that. So we're going to get a species produced in this family every few years. And it's also interesting that this occurs at the family level. Now, you probably see some similar pattern at the genus level, but you probably wouldn't see this at the higher levels when you mix these families in, because the curve, the slope of the line there, is going to be different. So it's interesting that within 700 years, you're going to have horses separated from donkeys, which that's the first split that we have. Meaning that's very likely, considering the rest of the time scale, that split occurred in the 700 years easily. Because remember, we only have 4,500 years to work with here. The same way for the flocks and the herds, the cattle and the sheep and the goats. That's one of the first splits that we see. Meaning, again, that it's very likely that this occurred not only in the first 700 years, but probably the first 200 years. So 700 years after the flood, as the Ice Age was finishing, we see that there have been just at least a few speciation events, most likely more. But so far from this information, we can only assume that there's been a few at least. But overall, the timeline fits with what we expect to see. So it doesn't prove the biblical model, but it is strong support for it. If we knew the mutation rate, we'd get more information out of this. But let's try to take this further. Let's look at the deer. Same slope. Well, not the same slope, but we still have the same linear pattern. Now, these are going to be different slopes. And so when you compare this to this to this, you're going to get vastly different patterns, if you get a pattern at all, which most likely you wouldn't. But we see the same linear slope pattern in all these families: dolphin, wolf, foxes, common bats, chickens and pheasants, salamander and newts, venomous snakes, tiger sharks. All these biblical kinds have this pattern. So this gives us another way of determining what is or what isn't a biblical kind. This also gives us information about the mechanism of speciation. And so we can not only see how things produced in the past, but predict what we'd expect to see in the future. Assuming, of course, that mitochondrial mutation rate is constant. This also shows that while the rate is constant of new species appearing every few years is constant for the family, it's not constant for a species. Meaning every time a new species is produced, that species' ability to produce another species is lessened. So that means whatever is allowing this animal to produce new species is reducing. And that basically points to the mechanism behind speciation, going from heterozygosity to homozygosity. If the evolutionary model was true, mutations occurring and then at random allowing for new species to form whenever, you shouldn't see a speciation decreasing in any species. You should see it remaining constant, if you expect it to be constant at all. But you should see that there's no change. Yet, that's not what we're seeing. We're seeing that every time a new species is formed, it's far, far less likely for that one to occur in the same time period. In fact, it's going to take longer for it to branch off. But overall, the rate is constant. This is pointing to another piece of information pointing to the fact that God put a lot of heterozygosity, a lot of different genes in us. And as you get separation of these organisms, this one species, this one biblical kind, population goes here, population goes here. They're going to have different genes in them. And simply due to statistics, to randomness, some of those genes are going to be bred out. Not because of natural selection, not because of changes in the environment, but simply from just random statistics. You have a small amount of genes in this population here, they're probably going to be pushed out. And so this population and this population now have different genes in them. If they were to reintegrate and rebreed, then you get the genes shifting around again. But this is what we expect to see. And this is exactly what we see with these different species. So when you look at zebras and horses, the imperial versus the domestic horse, and the plains versus the domestic horse, you see that there's 26 million or 28 million differences between these comparisons. Of all these differences in these species, only 2 million of these differences are heterozygotic, meaning that for the vast majority of differences among species, it is due to homozygosity. It is due to a separation of these genes. So the gene in the horse isn't in the zebra, and the gene in the zebra isn't in the horse. We also see this in house cats and tigers. 43 million DNA differences as far as base pairs. Yet only 1.5 are heterozygous, meaning the rest are due to homozygosity. Same way with the fin whales and the minke whales. So all this is pointing to how we get these species today, pointing to the fact that God put all this information in us, and as things separate, we get genetic differences. Now, as far as the horse goes, we know that since stripes are dominant, that when the horses broke off, the gene for stripes probably wasn't big in that population and eventually was lost. That's why horses don't have stripes. And while it is difficult, we can use the biblical mentioning there of horses and donkeys, and then donkeys and zebras splitting off by 1400 BC, because Romans were aware of zebras, and that fits perfectly with what we see in the genes. And so a speciation rate of the horse would be one new species every 640 years. Now this isn't taking into account all the extinct horses. There are a number of different horse species that we have in the fossil record. The problem is that we cannot access their DNA. We can see that there's DNA there, but it's like finding a book that has been buried for a while. You know it's a book, but you probably can't read it. And so we can't really compare it that way. Maybe in the future we'll figure out a way of doing that, but for now it's not likely. And that's basically what I said just there. Another thing of note is that a lot of families have very, very few species. In fact, while we went over some of the most species in them, many have less than 10, if not less than 5. And that is interesting because assuming that God selected animals on the ark based upon their heterozygosity, based upon their closely genetic relationship to the original creation, which that is an assumption. We know Noah was selected because of his righteousness. We don't know why or how God selected the animals that he put on the ark. But assuming that, that means that there had to have been some sort of extinction event early post-flood for these families, these biblical kinds that have few species today. And that's because if one of these floods, one of these volcanic eruptions, or simply overhunting or over-competition killed out one of the species of the biblical kind that had a lot of that heterozygosity in it, then that heterozygosity, that differences as far as genes, is lost. And that would explain why some species, like the mice, have all those different species that were produced not from human breeding. We have the bovine family, bovine biblical kind, producing all those many different species and breeds from human interbreeding. But we have some, like the horse, that even though we do produce a lot of breeds, don't have a lot of species. So there was likely an extinction event early in the horse lineage. But all this can fit both models until we go to the next step. So, do we see any new species forming today? That is a question, since prior to the 1800s, we didn't really travel the globe nor compare animals. In the 1758, we only had 154 of today's known 4,500 species of mammals.
5,400.
Thank you, 5,400. Sorry. That's 97% unknown. 100 years later, 178 of today's 5,400 species of mammals known, but 80% of those were rodents. Even 30-some years ago, we still were several hundreds mammal species lower than what we are today. And we still see tens to hundreds of new mammal species produced each year, or found each year, documented. Now the question is, are we finding these species, or are they forming? Now on evolutionists will say, no, we're just finding them. We're getting down, because the rate is reducing as far as new species that we're finding? Potentially. Or are we getting to the actual production rate? Since we're finding all the ones that were out there originally, but now when we look back again, oh, there's a new species here. We didn't miss it, it just popped there recently. This is hard to separate because you'd have to monitor all the animals all over the world. But just like the nuclear DNA, looking at the whole picture is difficult. It's much easier if you look at a small, localized section of it. And that's exactly what occurred with Darwin's finches. So since the 1970s, this couple here, Peter and Rosemary Grant, have spent a good portion of their life on an island just basically bagging and tagging. They would catch them, take DNA samples, tag them, and do very in-depth study. Not only on the creatures themselves, focusing on the medium groundfish on Daphne Major Island, but also the weather. And you can see that changes in the weather produce changes in the beak size. And a very small change of 1 millimeter thickness basically results in a change of diet. So when you have these climate events occurring, you're going to see a massive change, or a quick change I should say, in the beak size. Meaning that this is most likely not due to breeding, but due to epigenetics. Which means that Darwin's whole basis, as far as using that for evolution, isn't a great fit anymore. But that's not what we're focusing on here. Some time ago, there was a male large cactus finch that migrated and mated with a medium ground finch. Mated with a few of them. While most of the offspring were absorbed into the medium ground finch population, some did not. This was mainly due to their father's mating call not being selected for. Birds use mating calls to select mates. At least these birds do. So they interbred. And this couple here not only noted it, they got all this information together and they said that this is a new species. By all accounts it is. They named it Big Bird there. You can see a picture of it on the right. So it is an interbreeding producing a new species, but still it's a new species. So there are 11,000 bird species alive today. Now this isn't biblical kinds. This is just everything that we'd call a bird, their total number of species. Now, from the biblical approach, we would expect that 11,000 bird species divided by 4,500 years, meaning that we're going to have a new bird species every 2.4 years, basically. So every 2.4 years, 2.5 years, we'd expect a new bird species to appear somewhere on the globe. Now, the evolutionary model would predict 11,000 bird species divided by 35 to 56 million years, since they can't agree when birds actually appeared. You're going to get one new bird species every 3,200 years. And so previously we had 18 Darwin finches. Now there's 19. So let's take these expectations. Remember, those are global expectations. And look at what the data we have. So these birds were first discovered in 1837. One new species in 181 years. Remember, this is a localized rate, not a global rate. So that means to get this localized rate to a global rate, we have to multiply 1 divided by 81 times 11,000 birds. So the evolutionary rate would be 0.0003 new bird species per year. That's what they're expecting. The biblical rate, 2.5 basically. The observed rate is 3.4, meaning not only does it not match with the evolutionary rate, but it's on the opposite side of the biblical rate, and really, really close. Now, this was predicted by a Dr. Nathaniel Jeanson, I believe it was 2016 or 2017. 2018 is when this species was announced. And I just can't get over the irony that this is with Darwin's finches. So the same thing that he used for his model is what is a big piece of information supporting the biblical model. Remember, predictions make good science. If you can get predictions, then you have a good model there. May not be exact, but it's really close. And this is extremely close to what he predicted. It's like predicting a Super Bowl winner, not only the team, but also the final score, and being off by a field goal 50 years later. That's a good prediction. And so we have all this information showing not only how we got all these species today, but how the genetic information is produced in these species, and it all matches the biblical model. Now, there's a lot of information that we can go into with this, and a lot more studies that can be done, but everything, the more and more in depth we get with genetics, is pointing to the Bible being accurate in everything that it says. Now, I know this has been very, very heavy as far as the science behind it. Genetics is not everyone's cup of tea, and I'm sorry for that. And perhaps it's because of my own education that I was really excited about all of this stuff, because it's really neat, it's really cool, it's just earth-shattering for the evolutionary model anyway. And so, I'm apologizing if that isn't your cup of tea, but it is good information to have. Now, from this point on, we will be focusing more on archaeology. We'll be touching a little bit about genetics and stuff like that as it comes into play. But this is going to be a lot more about show and tell versus here's this equation, here's this formula, and going on. So I'm hoping that if this first portion of the lectures, first portion of the class wasn't your cup of tea, that this next one will be far, far more interesting, since one, everyone likes pictures, and two, everyone likes history. At least that's my experience. Any questions coming in? Any comments? All right. Well, that's it for today. Next time we'll be focusing on man and what he's doing in this post-flood world. Thank you.